on the line?
So we have everyone in the CMDI office, and Janet, Sabine, and Emily, and myself, and we can introduce. We have Charlene York, and we have Lindsay Swanson, the genetic counselor for Dr. Begg, and Sarah Foy. I don't know if she's audio. I see her in the presentation screen, but I don't audio.
Hello.
Great, so we're just going to go around and introduce ourselves. Janet's going to start off first.
Hello, I'm Janet Young. I am volunteer, CMDIR office volunteer.
Hi, Sandra. This is Sabine. You remember me.
Hi.
Hi.
Hi, Sandra. My name's Emily Pinu. I'm also a volunteer at the CMDIR.
This is Rachel. You know me.
Yeah.
Charlene?
Yeah, this is Charlene York. My husband and daughter were genetically tested for the collagen 6, A3. So that's how we got our diagnosis, was with genetic testing.
Charlene does lots and lots of work for CMD.
Yep. Volunteer too.
Lindsay?
And I'm Lindsay Swanson. I'm the new genetic counselor for the Begg Lab at Boston Children's Hospital.
Sarah, are you on the call?
Yes. Hi, this is Sarah. Yes. I am a mother of a son with a congenital myopathy, and I also have a leadership role on the Congenital Muscle Disease International Registry.
I think that's it. On to you, Sandra.
Okay, great. So, yeah, again, I'm sorry I'm late. It's been a hectic day. So I am the genetic counselor working with Dr. Bodeman here at the NIH. And genetic counseling is kind of a relatively new field. We are not doctors. We have a graduate degree my graduate degree is in human genetics, but they also have specifically programs for genetic counseling. My bachelor I actually did back home where my family's from in Holland, and I was pre-med, and I did a lot of the laboratory courses, but I hated everything that had to do with practical issues, so I liked all the science and the medicine, but I could never actually work in a hospital and do an exam or work in a lab and do blobs and pipetting and all those technical things. And then, so genetic counseling was perfect because it's a mixture of the science, so the genetics, the medical genetics, learning about all the different diseases and the genetics of it, as well as just working with the patients, but not really having to do any of the exams or lab technical testing. I just get the results and then discuss it with the patients. So when I graduated from Sarah Lawrence, I moved to Northwestern, where I worked with the MDA clinic and the ALS clinic, and I briefly went to California, and now I've been here with Dr. Bodeman for a little over a year, kind of like wearing different hats, clinic coordinator / genetic counselor, and then we do the traveling clinics together with Anne to help local teams with the genetic diagnosis. So these slides are just I think a basic background of genetic testing and the importance in the CMD patient population. So we all have thousands and thousands of genes, and these genes are very important. They all have different functions, but they're really important in our development and function. So the genetic test is the analysis to try and see whether or not there's any alterations that could explain some of the symptoms that some patients may have. And I'll go back to this. A disorder can be genetic, but it may not necessarily mean that it's inherited. So this is where the family history comes in, and we'll go over different ways in which the family history can guide us or can give us clues as to which genetic testing we want to order. So when we order testing, there's a lot of different information that we have to take into account. So of course you want to know, what does the neurologist see, what are the patient's symptoms, in addition to as many other findings that could give us clues as to which genes you want to test for, because there are so many genes that the best thing is to do a targeted approach. So sometimes biopsy findings can be helpful. Sometimes imaging data, either on MRI or ultrasound, can be very helpful because there are specific patterns that we can sometimes see that are kind of very specific for a subset of disorders. However, you have to be lucky to be able to work with someone that can recognize that or that knows about all those very specific patterns. And then any additional clinical data that can help us guide as to which category of genes we need to look at, whether someone has more of an intrinsic muscle problem, whether the muscle problems is related to nerves, or whether there's any heart involvement, just to give us guidance as to which category of genes we have to look at. And then where I come in, usually before we see a patient, is with the family history information to see if there's any clues in the family history. So we just went over this. When we take a family history, we ask about everyone in the family to try and see if there's obviously anyone that has similar symptoms. Sometimes it's not necessarily that someone has an identical presentation as the affected patient that we see. It is possible that maybe a parent only has very flexible joints or some very, very mild weakness that they themselves may not necessarily pick up on, but if they go through a neurology exam, the doctor can pick up on. Sometimes what we'll look at is a heart condition in which maybe a parent has an early onset heart condition, and then the child, it manifests itself as a heart condition and maybe also muscle disease. So we're just trying to see if there's any clues that can guide us. Sometimes what's very helpful is to know where the family is originally from, ethnicity-wise, because in some disorders are more prevalent in some ethnicity, so that can give us clues, which is why we always ask about that. However, it's very possible that we ask all these questions about the family history, and then everything comes to everyone's healthy, or there's non-related issues like diabetes or later onset cancer. So it's very common, and usually families come in having already investigated themselves everything in their family to try and see or to try to come to any kind of explanations of why all of a sudden this happened out of the blue in one of their kids. So when we take a family history, this is what the pedigree looks like. So we have squares are for boys, circles are for girls. So this will be our affected, the affected boy with his older and younger siblings. Then we have parents, dad's brother, dad's parents, mom's sister, mom's parents. This is how we draw everything out. And then this is where clues can be very helpful, or where it's very easy to illustrate if there's anything that comes up that's of interest. So this first pedigree here shows definitely that that the muscle disease is inherited from generation to generation. So both males and females are affected, and you can see it's passed down from one generation to another generation. It is possible that over time the disease gets more severe. That's known in certain conditions, but it can also be possible that it kind of stays the same. It was just never really diagnosed. It's also possible that it's passed on through the females. So females are unaffected, but the males show symptoms, which is an X-linked disorder. So these are patterns that's a little harder to pick up on. But the key finding is only boys affected and no females affected. But most commonly what we see in our clinic is that families come in which both sides of the family are completely healthy, and then there's one affected individual. Another possibility, so there's different ways that could possibly explain that. It's either something that happened at the time of conception, so neither one of the parents has anything that they carry in their genetic material, but it just happened at conception. Sometimes things happen. They happen all the time. It's just a matter of fact whether it happens in a critical region of a gene that's associated with muscle disease. But it's also possible that both dad and mom are a carrier of something that they both pass down to the child in order for the child to be symptomatic. So these are things that, based on family history, we can't make the distinction whether or not this is something that happened, what we call de novo, or spontaneous at conception, or whether or not it's something that both mom and dad carry, in which case there is a significant risk for it to occur again, and for another child to maybe be affected in future pregnancies, which is why it's really important for us to figure out which one of the two possibility is what causes, or what could cause the symptoms, whether it's inherited through mom and dad, or if it's something that happened at the time of conception. Okay, so this is one of the few basic science slides about genetics. So what a gene really is just a strength. It's a code of letters that is an instruction code that tells our body how to work and function properly. So it's really important that the instruction code is written correctly. We have 2 copies of every gene, one's from mom and one's from dad, and they're located on chromosomes. So this is what our chromosomes look like if we line them up nicely. So they come in pairs, one's from mom, one's from dad, and on these chromosomes we have our genes that are kind of in like condensed packages. So what we know is that this instruction code is important. It tells our body how to function and develop correctly. And certain genes are specifically involved in muscle cells and muscle development. So there are specific genes that are necessary in order to make the muscle membranes, to make the muscle cells and the membranes strong and linked together. So it's very possible, if there's a change in this gene or there's a problem with the instruction code, that the muscle membrane strength is reduced, or that there's more muscle breakdown. We also know that sometimes proteins can be involved not just in the muscles, but also in the heart or other parts of the body, which is why certain conditions are more of a multisystemic disorder in which the instruction code is important for more than one organ. Another example is the brain, in which sometimes it's not just a muscle disease, but it could be a muscle-brain-eye phenotype. So the instruction code, as I said, are letters. So it's really 4 letters that are just on a string. So thousands of letters that the code is really important. So the letters are repeated in a specific order, and the body reads that order and then translates that into a specific protein that's sent on their way to do their specific job. So this is another schematic of it. So you have your cell. In the middle of the cell, you have your chromosomes, and that's really a condensed string of letters of the DNA that are positioned in a specific way for the body to read and then produce the proteins that really are the ones that do the function, that are sent off their way to do the specific work that needs to be done. So there's specific types of genetic testing. This is really a basic outline of the different types of testing that you may run across. So the first one is cytogenetic testing, which is really the first initial basic screen that they do. Usually by the time you see kids here, they already have the karyotype. That's what it's called, done. So they will run a karyotype whenever they see a child, usually very early on, that has any kind of multiple anomalies, developmental delay, or any kind of unknown presentation. So the karyotype, really what it is to make a picture of the chromosome. So it's a very crude picture in which they line up the chromosomes correctly. So this is what a normal karyotype would look like. This would be all the chromosomes paired up and numbered, and then XY is boy, 2 X's would be a girl. So this would be a healthy karyotype. And then here we see a karyotype of someone that has Down syndrome, in which instead of 2 chromosomes, 21, we would see 3. So usually this is already done by the time you see the kids. Sometimes it's applicable because it can also pick up on either small deletions or small duplications. So this is just what's visible by the eye, what you can see. in genetic counseling school, one of the first things you have to do was cut out these chromosomes and line them up as a karyotype, which is definitely not a fun thing to do, because for me they all kind of look the same. But here it will be a deletion on chromosome 4. There's a little bit of white stuff missing that's on this chromosome, but not on this one. So this is important because now what we would have to do is figure out which are the genes that are located here in this white stuff in this portion of chromosome 4, and are any of those genes, could they explain the symptoms that we see in the baby? But as I said, usually by the time we see the kids, this is already done because this is relatively easy for, it's very standard that doctors order this whenever they see a child with unexplained symptoms. So the second test that they usually do by the time we see kids is an array. So an array is also very, very non-specific. All it does is it looks for whether or not there's any extra or missing genetic material. So it looks at whether there's, when you look at the karyotype, you look at chunks of DNA that's visible with the eye. When you look at the array, the machine does all the work for you, so it's a lot more specific. So you take the patient's DNA, you blot it, and you they use all kinds of special laboratory techniques, and then with the computer it spits out whether or not there's any missing DNA here. So it's a lot more sensitive, and it can pick up very, very small deletions or duplications. And sometimes it's very, very helpful for us if there's only like 1 or 2 genes missing or extra to get the diagnosis. But again, this is a very, very non-specific test, so you don't really need to have a specific disorder in mind. It's just to see if there's extra or missing DNA. And sometimes it can guide us towards, if we know that there's a specific area of the chromosome missing, we can see which genes should be located, and then which genes are the ones that are important for the specific symptoms that we see. Okay. Then, well, we just went over this. The direct sequencing, this is the one that we usually do in our clinic. So this is when we look at a patient and we put all the information together. So we do the physical exam. We look at the MRI imaging. Are there any specific clues there, any patterns? For example, with collagen, we look at the patient. We see the specific signs of weakness that we see in most kids. We see the bendiness of the finger. We see the contractures. We see the specific ultrasound findings. And then we're like, okay, we're pretty confident this patient has a collagen 6 disorder. Let's send off for genetic testing to look specifically at the collagen 6 gene. So the direct sequencing, what happens is that the laboratory is going to read through the letters one by one. So they read through the instruction code, and they know what the code should look like, and then they read through it to see if there's any misspellings in the code that we know can cause disease. So here, this will be the normal code, but instead of an A, this patient has a G, and we know that every time we see the G, we only see it in patients with myopathy. So we're pretty confident, okay, the genetic testing confirmed what we were seeing clinically, and this is the specific result of the testing. What's also possible is instead of one single base pairs change, is that there's 3 or 4 chains, or they're missing, or they're extra. Just anything can happen that can mess up the instruction code so that the body doesn't really know what to do with it, and the right proteins are not produced anymore. So this is the direct sequencing, where you have in mind what you think the patient has, and you tell the laboratory to look specifically for that one gene. What is possible, what makes it a little tricky, is that we all have changes in our instruction code. So we all have variations in our genes because that's what makes us unique. So for us, sometimes we know this is a specific change as a variation because we've seen it in all these healthy individuals, so we know that, you know, if this G changes in an A, it doesn't really matter that much because 30% of the healthy population has that. It's just a random variation. So sometimes there's some extra investigation that needs to be done when we get our genetic testing results back to see whether or not the specific spelling mistake is just a random benign variation or if it's something that's associated with disease. So what we can do then is to test the parents. If it's something that's inherited from the mom and the mom is completely fine, then most likely it's a benign variation. And sometimes what happens is that the lab just has to test additional patients and additional samples to reclassify one of those changes over time to say, okay, we've tested more and more patients, and we see consistently that this change comes up with the patients and not necessarily in healthy controls. So even though 5 years ago we said that this was a variant of unknown significance, now with more data available and more testing that we did, we know it's disease causing. Or maybe they tested all these healthy volunteers, found it in healthy volunteers, and now know it's just a benign variant. So sometimes it just takes longer to really get to an answer with the testing. These spelling mistakes are in every single one of our cells because kids and families always ask, so why can't we just go back and correct it? Which is a great question, and hopefully in the future we will be, but the whole gene therapy, it's very tricky, as you can imagine, because every cell has this spelling mistake, and this is usually there at the time of conception, at the time of birth. So it's really all over and not necessarily just in the muscles. And as I said, these mutations, these spelling mistakes can happen spontaneously, and they always do happen. Like at the time of conception, there always are spontaneous mutations. Usually they happen in our junk DNA, the DNA that you don't really need, but it's very possible that they happen in muscle disease or any disease. So it's not uncommon to have spontaneous mutations. It just is, it's just a coincidence whether or not it's in our so-called junk DNA or in DNA that's really needed for development and for function. And then again, sometimes it is inherited through the family, either from generation to generation or in a situation where both parents are a carrier. Okay, so the benefits of the genetic diagnosis, obviously it confirms the clinical diagnosis. So if there's a clinical suspicion, or even sometimes when when it's not really clear which way to go, like with the alpha-dystroglycanopathies, where it's not always as clear because some of the genes may overlap, it confirms the diagnosis. It will provide information for management because not all, because there's a difference. We know that some genetic mutations in muscle disease are to be involved with the heart, so we would know that we need to check the heart. However, in some situations like the OLIS, we know that the collagens are not necessarily involved in the heart, so that's one less thing that we need to worry about, one less doctor visit for the kids to do on a yearly basis. The risk itself for genetic testing is very low because it's just a blood sample. So that's by itself. It's a relatively easy test. It's not as invasive as a muscle biopsy or some of the other testing or spinal tap. It's very important because once we know what the underlying genetic cause is, we can provide recurrence risk information to the family. What are the chances if they want another child for this to happen again, or what are the chances for the affected individual in the future to have an affected child or a healthy child. In some situations where we're able to pinpoint what the underlying genetic cause is, families can do, they have the option of prenatal testing if that's something that they want to discuss or learn more about. So it opens up a lot of options there and information about recurrence. And then, of course, as we move forward with clinical trials, it becomes more and more common that an underlying genetic diagnosis is needed in order to enter the trials. So it's good to have a complete picture available, of that complete documentation of the patient's medical information. The limitations, it's not always as easy to get a genetic diagnosis. It may require multiple tests, and it's not uncommon for patients to come in with already 5, 6, 7 genetic testing or genes sent for genetic testing, and they're all normal. It may not identify a mutation, so you can send off one test, comes back normal, then you send another test, comes back normal. So sometimes you really have to be persistent, or maybe get additional clinical information or a second opinion from someone that's maybe an expert in that field, so they can guide towards more targeted testing. It is tricky because sometimes it does not necessarily help that much with prognosis and severity, especially in situations where it's more of a rare genetic cause in which there's just not enough information out there. Maybe there's 5 other kids reported with the same underlying genetic cause, and they all 5 have a very, very different prognosis and very, very different cause of disease, in which case it may not be very helpful to really provide any prognostic information. And then it can be a very costly test, and insurance may not always cover it. So sometimes it's multiple battles with insurance in order to get it covered, or go the research route, and that may sometimes take a little bit longer to actually get results. And then it's possible to get unexpected results. So like the variants, in which we just don't really know what it means. Is it a benign variant, or is it disease-causing? Should we wait till we get more information, or is it just a red herring, and are we really missing what's going on? So it may not always give a conclusive information or any, like sometimes you kind of get stuck with the results, so you don't really know what to do and what to make out of it. So sometimes, so this is just a picture that goes over that, in which you're not always really sure what a result really means and how to interpret it and what to do with it. So how to get tested is really relatively easy. It's just a blood sample through either the neurology. In some hospitals, you have to go through the genetics department. Where testing is ordered, it's important to know what the method is. So whether it is a direct sequencing or if it's a panel, there are certain labs at this point that have specific panels in which they have some of the common CMD genes, so the collagen, alpha-dystroglycanopathies, which is great because it captures a lot of genes at once. However, they're also very expensive, so if insurance doesn't cover it, it may be better to go by the one that's most likely and then slowly go down the list. As I said, it's definitely expensive, and insurance can be a battle. It can take months for results to come back, and then it's always important to know what are the possible results that we can get back with the testing, like what should we expect, and not always, patients don't always understand that a result can be an intermediate result, and that it's not always a clear yes or no. So it's good to have those expectations lined up beforehand. As I said, how do I interpret the results? It's always good to sit down with the genetic counselors or with a neurologist that's familiar with genetic testing, and to keep copies. It's really, really important to have copies of the genetic testing results available, because it's very hard to track down, and then of course register. Additional info about genetic testing is really, especially here at the NIH, we talk a lot about clinical testing and research testing. Clinically, there's a lot of the direct sequencing, the direct testing is available on a clinical level. And we work with labs like Prevention Genetics and Emory and Athena. They do a lot of the direct sequencing of genes, and also they have panels. And then here at the NIH, we do a lot of research testing in which we can test for genes that are not yet reported. So maybe our lab or one of our collaborators recently identified a gene and it's not yet reported, it's not yet commercially available, so we will be able to test for that on a research basis. And then another test that's typically at this point still done on a research basis but may move more in the commercial field is exome testing. It's a pretty recent technology in which instead of looking at one gene at the time, so reading through the instruction code of one gene at the time, with the exome testing you read through all of the genes. So typically this is what we would do in a situation in which we've ruled out all the known genes, we don't really know what are the genes to test for anymore, and we feel that maybe the patient has a gene that's not yet discovered because the patient doesn't really look like any other patients that we've seen before. It's kind of atypical presentation, so we would consider doing the exome testing to see if maybe it's a gene that's not yet discovered yet. The exome testing, how it would work is we would screen all of the genes. So it's a big project. It can take 6 to 8 months, just the technical part of the project to read through the genetic material. And then we would compare it to both of the parents to see whether or not there's any variance in the parents, that maybe they're a carrier and then both pass down to the child, or if there's any variance in any genes that we know are very important in muscle disease that happened in the child that happened at the time of conception. So it's more of a fishing expedition, the exome sequencing, in which we, instead of going in a targeted approach, we just look at all of the genes and then kind of like zoom in on specific ones that could be involved in muscle, but not really with a specific hypothesis in mind. Hopefully the exome testing will become more available, in which the turnaround time will be a little bit faster, and in which there's specific interfaces in place that can help us with the analysis of the data, because you send off for an exome testing and then it comes back with just thousands of variants that you have to slowly work your way through. So it's a very labor-intensive test to do. However, it's also very successful in identifying new genes, and hopefully in the future it will just make those genetic diagnoses a lot easier, in which we don't have to test one gene at the time, wait another 3 months, test for another gene, wait another 2 months, but we can just do the exome and then zoom in all the genes that we want to evaluate. So hopefully the whole genetic diagnosis process in the future will just be streamlined because of the exome testing. The one thing to keep in mind with the exome testing is because you look at all of the genes, it is possible that you're going to find information that you don't necessarily want to, or that's not necessarily what you want to look for at that time. So there's a lot of information in our genes. You can think about predisposition to specific cancers, or just any kind of dementia, any kind of information in your genes that maybe when you look at a 5-year-old in your clinic because of muscle weakness, you don't necessarily at that time want to deal with. So there is a lot of ethical issues involved with the exome testing, even though it's very, very, very promising and hopefully will really help with the genetic diagnosis in a lot of our patients. Okay? Any questions?
Yeah. How does the exome testing compare to the sequencing of the whole genome?
So the exome testing, that's a good question. I didn't specify that. So the exome testing only looks at the parts of the genetic material that's used for the proteins. So it's the part of the material that the body actually reads for protein production. And the whole genome looks at everything, so that also includes some of the junk DNA that we don't necessarily use for protein production. So the exome is a more efficient way of doing the testing.
Okay, because I've been heard these reports on the radio that a lot of people now start doing, having their genome tested or sequenced.
Mm-hmm.
and that the prices are coming down, that soon, I mean, one company had a bargain for $999. You can have your genome sequenced.
The data.
I know, exactly. And
interpretation. Do you, Lindsay and Sandra, do you want to just talk a little bit about kind of, because I think this would be very interesting for the group to hear a little bit about some of the ethical considerations of whole exome or whole genome sequencing when now you're not really going after, I mean, you say you're going after trying to identify the cause of the disorder of the person standing in front of you, but clearly you're getting sent back information that could also inform you about other diseases that they've inherited or they're at risk for. And now if you have parents who are consenting for their child to have whole exome sequencing, kind of what are some of the ethical considerations that one should be aware of, and what kind of worms could this potentially open up? Yeah, no, that's definitely a great point. It is something that, to be honest, it's not anything that's really new. With genetic testing, you can always come up with things that you don't really expect. It's just with I think with the exome testing, the exome sequencing is going to happen a lot more. One of the things that you can think about with the direct testing, and that has happened with one of my patients, is that you test the patient clearly has a recessive disorder, so inherited something that from both mom and dad. Mom and dad wanted to know what the recurrence risk is, so to complete the picture, you test mom, you test dad. Both have to be a carrier, because that's the only way to explain that the kid has a recessive disease. Mom's a carrier and dad's not a carrier. And then you sit down and talk to mom, and mom tells you that, yeah, dad's not really dad. So these are issues that I feel these ethical issues have always been part of genetic testing, and it's always something that with a patient I discuss, especially since that happened, I discussed that additional information can come up when you do genetic testing that you may not expect or you may not want to hear or be ready to hear. But yes, with the exome testing, there's a whole different discussion about we're going to find these changes because we all have changes in our DNA. And then what are the changes that you want to hear about, and how are we going to discuss this with a 5-year-old talking about maybe there's a risk for breast cancer that's very different than when you talk to a 30-year-old. So at this point, we have very strict regulations by our institutional review board here at the NIH, in which we can only disclose any kind of information in which we feel it can impact their health. So if we find something that that's actionable, so if we find something that an increased risk for cancer that they can screen for the patient and their family members. But if we find something that's just something that you can't do anything about, then we're not allowed to tell the family. But again, it's tricky because this exome testing is very, very new. So every hospital and every research group has their own guidelines and regulations about what they can disclose to the families. And I don't know how they do it. Lindsay, maybe you can comment on what your regulations are. But I think it's different at every institution.
Right. Every institution and every different clinical laboratory that's offering whole exome sequencing varies in the way that they've set up their informed consent, and that's why it's recommended that individuals pursuing exome sequencing meet with a genetic counselor or some genetics provider that can sit down with these families and explain the implications of exome sequencing and what may come with those results. So yeah, so right now we're kind of pursuing things on a case-by-case basis since we've just started doing exome sequencing on a small group of the BEGS lab participants. So.
Great. Lindsay, do you have anything to add to what Sandra has presented, or does anybody have any additional questions?
No, I don't think so.
Emily has a question. Hang on.
You mentioned that you can determine based on looking at mom and dad's DNA if they are likely to produce another child with the same or similar disease. are you able to tell them or to, like, do you then refer them to, like, family counseling to, like, so they can have children without the disease, or is it just kind of chance?
Yeah, no, that's a great question. So there's in genetic counseling, there's kind of different fields. So I work with specifically in the neuromuscular field. So you have an whole other group of counselors that work in prenatal counseling. So if we identify what the underlying cause is in the patient and we know that there's a recurrence risk for additional pregnancies, I would always recommend a visit with a prenatal genetic counselor just to sit down and talk about options. Just a discussion about what the options are, and sometimes parents it doesn't really, it's very personal. So in some situations, yes, it's possible to test the pregnancy either through a CVS or amniocentesis to see whether or not the pregnancy is affected. Or what's also possible, and it's has happened a couple of times, in which the couple opts to go through IVF and then test the embryo, and then would only implant the embryo that's not affected.
Okay.
So these options become available. However, none of them are necessarily ideal.
Right.
But they're definitely options.
Okay, thank you.
I have a question. What happens if the patient comes back negative for any genetic mutation, but yet the skin fibroblast shows some kind of CMD involved? Is it just because the gene mutation hasn't been identified yet, or do they follow up with another DNA test?
With the skin fibroblasts, are you referring to the collagen staining?
Yes, yes.
So usually, yes. So collagen is one of the few disorders in which you can get some extra information by looking at just the collagen protein in the skin cells to see whether or not there's a problem in the skin cells. The collagen testing itself is tricky because we know that sometimes with the collagen gene, there can be some mutations that are kind of hidden. So you have to do additional testing that a commercial lab may not always be able to do, but we can do it in our lab to try and hunt down those hidden mutations. They're either very, very deeply in the gene. There's just more of a technical issue with the testing. So if the patient really clinically looks like they're affected with a collagen disorder, and then the fibroblasts, the skin cells, show that there's a problem with the protein, then really it's a matter of, in our lab technician will try and hunt down with various technology and testing, try and find what the hidden changes in the collagen gene.
Okay.
In general, the ones that if there is a problem in the skin cells with the collagen, it is typically just a hidden mutation. If the clinical lab can't find it, it's a hidden mutation within the collagen gene.
Okay, okay. Although this person's siblings has the collagen 6 issue, she was tested negative, but yet she has the symptoms of it. She hasn't had the skin done yet. We got to get it. This is just a family member that we have to get out to NIH on this.
Exactly, because it's, again, even with the collagen 6, the classical symptoms of the disease are relatively, if you've seen it, relatively easy to pick out. But there's, as you know, all these variations in which it can overlap with other disorders as well. So it could be that it's a secondary cause, or that there's something else going on that may be unrelated, but have an overlapping presentation.
Okay, thank you.
Great. Well, Sandra, thank you so much for such an informative presentation, extremely helpful, and thank you for allowing us to tape it. The plan is to post this and prior presentations on both the Cure CMD website and the CMDIR to enable the community to access these as educational resource material.
Sounds good.
Great. Thanks again, and have a great trip to Australia.
Thank you.
Okay. Talk
Bye.
to you later. Bye-bye.